All right, everybody. It is now 12:02 and we're going to begin the webinar. This is the first webinar in our OWC Innovation Showcase Fall Series. We're here with McMaster University and it's entitled Integrated Approaches to the Development of New Water and Monitoring Technologies. But before we start the webinar, we just want to talk a little bit about who we are as the Ontario Water Consortium. The Ontario Water Consortium is Ontario's only sector support organization dedicated solely to water innovation. We bring together researchers, companies, municipalities, and governments to drive collaborative R&D, pilot testing, and real-world technology adoption. We bridge the gap between research and market-ready innovation. And while there are many innovation organizations across Canada, none are focused specifically and exclusively on water like OWC is. OWC's collaborative model delivers measurable results. Over the past decade, we've helped mobilize over $90 million in project funding, supported or created more than 350 industry jobs, and advanced nearly 80 water technology projects. We've partnered with more than 100 academic researchers and helped build infrastructure, including wastewater test beds in London and Guelph. We do this by connecting innovators with adopters and aligning the right funding, partnerships, and support to move solutions forward. And before introducing today's webinar, we'd also hope you join us again for the rest of the fall webinar series. On October 2nd, we'll hear from Toronto Metropolitan University, showcasing research and tools for quantifying and predicting greenhouse gas emissions from wastewater treatment plants. And then on October 8th, the University of Windsor will present interdisciplinary approaches for restoring and protecting freshwater ecosystems with a focus on biodiversity conservation across the Great Lakes Basin. I will drop the registration link in the chat in just a moment. All sessions are free to join. Please also note the QR code here that you can use to subscribe to OWC's newsletter. Now, I'm very pleased to introduce Dr. Herb Schellhorn, Professor of Biology at McMaster University, to begin today's webinar, once again entitled Integrated Approaches to the Development of New Water Monitoring Technology in Ontario, the MacWater Diagnostic Research Initiative. Dr. Schellhorn following Dr. Schellhorn, we will hear from Dr. Faizan Saleem, who will showcase innovations in microfabricated sensor technologies, and Dr. Ravi Selvaganapathy, who will provide an engineering perspective on sensor design and system integration. Herb, the floor is yours.
Great. Thank you very much for that introduction, Robert. And I'll just share my screen here. If a little hiccup. Can you see my screen?
Not quite yet.
No?
It may be loading, so this can take a moment.
Share entire screen. Okay. Now you can see my screen?
Yep, something's coming up. I can see your screen.
Okay.
Yeah.
Great. Well, thank you very much, Robert, for that introduction. As you said, I'm a professor of biology at McMaster University, and we started working on water technology about 15 years ago, and this was a new thing for me. We began with a partnership with Niagara, which was really helpful in giving us some seed funding for beginning an initiative. And we didn't have any external interactions before 2008, but after that, we got that partnership with Niagara, which was really helpful. And it led to an Ontario Research Excellence Fund and allowed us to form the McWater Identity, which is an interdisciplinary group. And we recognize that if we're going to work as a team, we have to identify relevant expert people to help with this, and so it's interdisciplinary. But we also need external partners to help us work on real world problems. And it's certainly in Southern Ontario, that's a great environment for that, because there's a tremendous number of companies that are invested in this. Then were funded by several CRD Connects and Advancing Water Technology grants from Ontario Water Consortium. We've had a long relationship with OWC, and we're glad that they come to our meetings. We received a second ORF through Guelph through the NSERC Alliance program, which was, and then an NSERC Alliance grant that helped to support research more recently. And we've expanded our partnership with the Ministry of the Environment and Environment Canada to work on harmful algal blooms. So this has made us a very interactive group of people who can work with external partners, and funding agency recognize that. So first we had to develop an interdisciplinary team, which includes finding complementary expertise. Cooperation between science and engineering, I think, is essential. And here at McMaster, we're really fortunate that we have a lot of great engineers like Dr. Selvaganapathy to work with who are biology friendly or science friendly. So that's really helpful. And external partners, I get cold calls now to work on problems. And this is really interesting that we're working on problems we couldn't have imagined 10 years ago. But it's really helpful, not just for us, but also as a training environment for students and as a platform for future job opportunities. We have created combined training initiatives to support training in engineering and science, mostly to give engineers a little more science exposure and science students a little more engineering expertise. This was our initial diagnostic initiative team, and you can see that we have a lot of complementary expertise that we can bring to bear on biological problems. And many of these have experience in developing industry solutions. So our initial goal, and this still is true, we're trying to develop low-cost, real-time portable devices that can analyze real-world environmental water samples for biological and chemical contaminants. And we have this mission statement to differentiate what we do from other water-related activities like environmental monitoring or pure remediation. Ours is mostly of a diagnostic nature, and then it's important to differentiate from other groups. There's also science policy groups that work on water, and we're not really involved in science policy, although we do supply information to policy decision makers like public health. I mentioned this is an integrated approach to water monitoring, so we have to look at this in its entirety, and this includes end user interaction, collecting samples, accessing metadata, developing sensors and methods of analysis, combining this information into robust data sets that can be used for combining with satellite information or weather as metadata, and return information to the end user. And a good poster child for this approach is our current project with the Ministry of the Environment, where we're under a contract to assess lots of different types of technologies that can be used to estimate the impact of algal blooms that can help Public Health Ontario develop a more response to end users who are interested in knowing whether the water is safe with respect to algal blooms. So this has been enabled by several developments in key technology, and I'll go through this quickly. Inexpensive DNA sequencing has been a game changer. Semiconductor-based sensors and increased computation, and now AI are we're bringing to bear on water problems. So this is really useful. Our group has been a useful vehicle for bringing people together. We also recognize that this has lots of training opportunities for students, and that's what's shown in the outer ring in the slide. And so we try to address these training issues in our research and training programs that we've developed. Now, one thing that's unique, I think, about us is we have a meeting, a Challenges in Water Monitoring meeting that we hold at Innovation Park every year. It's attended by 6 funding agencies, including Ontario Water Consortium. And I think it's been great for us. It's a one-day meeting, so you just pop in, spend the day, meet a lot of people, and you learn how agencies like Ontario Water Consortium fund research. And we even have the Canadian Revenue Agency come and talk about SR&ED tax credits for research. And so they're trying to give companies money, and we find that a lot of younger startup companies are unaware of that possibility, and yet the benefits are greatest for them. So we have worked with them closely for almost 15 years now. So this is invitation only in the sense that we want to make sure that it's people who are interested in creating new strategic partnerships with industry. That's the goal. That's what I put out in my invitation letter. So if you're there for something else, you're at the wrong meeting. We focus on partnerships, and I think from almost every meeting we have formed some partnerships and some really interesting ones. And I don't have time to go into it, but they're out of the blue. People call me and say, we have this problem, we want to come to your meeting, and I send them an invitation letter. So this helps to create new initiatives to support research. Many federal funding opportunities now depend on partnerships, and so this has been a great platform for that. And it's recognized that water and wastewater monitoring are going to be a key to any remediation effort. So the basic format of the meeting, and I'm just pointing out that it's just one day. We have funding agencies, tells us what's new. We get an update from reporters, a keynote address, a great poster session for networking, some more update reports. And new in the last couple of years, we put in a McMaster technical forum that informs companies about the 10 central facilities we have on campus that can support water research, like a high throughput sequencing facility, the Canadian Center for Electron Microscopy, the Center for Advanced Light Microscopy. These are all important tools for us to use in addressing industry problems. And of course, we have a reception in the late afternoon where we have informal contact. So I'm going to bring me to the end. We have a website that describes our activities, and you can find programs of our past meetings at this site that shows you the kind of companies that have attended our meetings and the kind of talks that we've given. We basically, originally we had 3 objectives, and we still have that. That includes surveillance and bioinformatics, and that's a big part of what my lab does. And Faizan will talk about that in a couple of minutes. And we have sensor systems, and Ravi has been involved in that since he started as a professor, and that's really helpful. And information fusion, bringing things together and creating real time methods of collecting information is a final objective. So I think I'll end there. I'm going to turn it over to Dr. Faizan Saleem, who's a postdoc and has worked extensively on DNA analysis.
Thank you very much, Dr. Schellhorn. As Dr. Schellhorn has mentioned, my name is Faizan Saleem. I'm a postdoctoral fellow in the Department of Biology, McMaster University, and I've been a part of MacWater group for the last 5 years. What I'm going to talk about today is how we can use the innovative DNA technologies to improve the water quality monitoring by bridging engineering innovation, commercial impact, and training. So when we talk about improving the DNA water quality, what we actually want to do is we want to make it rapid. We want to make it more specific. We want to identify the contamination sources that are responsible for the water quality, and also we want to identify the harmful algal blooms, which has become a recurring problem around the Great Lakes, especially in the past 20 years. So in order to do that, let's start with the first objective, which is the rapid water quality monitoring. Now, in order for us to understand this, first thing that we need to realize that what are the conventional methods that are being used right now, which in this case, in water quality monitoring, we use bacterial culturing. It takes about 24 to 48 hours for the bacterial cultures to grow after the sample collection. Now, during that time period, which means beach managers or the public health agencies cannot make a timely decision on whether to open the beaches or the water for the recreational purposes or not. So to alleviate this, US EPA about 10 years ago introduced a DNA-based method which can provide or compress the timeline from sample collection to the acquisition of results within 4 hours, which means it can allow the decision makers to make the decision on the same day of the sample collection. Now, we tested this technology specifically in the context of Ontario freshwater beaches for about 3 years. And we found that it can be deployed, and it can be easily be used for the purpose of the same day water quality monitoring, and can be a potential replacement to the conventional water bacterial culturing or bacterial growth based methods. And if you want to read a bit more about our findings, you can look into the publications that I have on the slide. Now, the second thing, or the second objective that we have, is to make the DNA-based technologies more specific, right? In order for us to understand that, we need to understand the limitations of the current DNA-based markers that we have. So those are universal in nature. When I say universal, it means that all the bacteria have those DNA sequences in them, but there are minute differences which are targeted to differentiate one bacteria from the another. So that creates sometimes false positives and false negatives, which we don't really want, because this has to be robust. So in order to alleviate this, we have Dr. Rad Gupta's made in McMaster technology is called conserved signature proteins, which are proteins and corresponding DNA sequences which are highly specific to the targeted organisms. And over the past 20 years, he has identified thousands of these related to specific bacterial communities of interest. And we can use these to develop more robust qPCR or based methods or DNA-based methods for the water quality monitoring specifically, which we did. In a specific context of E. coli and Cryptosporidium, we used the conserved signature proteins to develop novel qPCR-based methods which can be successfully deployed for water quality monitoring. Now, this can be extended to ideally all the other pathogens or the bacterial communities of interest, and our lab is currently working on other organisms as well, including Fascicola and harmful algal blooms or cyanobacterial communities. Now, the third objective is, if you want to have a long-term preservation or sustainability strategy, you want to identify the root cause, which is the fecal contamination sources. Traditional methods can't really do that. They don't really provide this information. So we use the eDNA sequencing or DNA sequencing to simultaneously identify all the potential fecal contamination sources in Ontario freshwater beaches, which can provide a platform or a comprehensive profile that can be used to stop the influx of the fecal contamination from specific biological sources. And the fourth one is the most common one that you have heard in the news clippings every year in summer. There are harmful algal blooms we have to close down, because of which we have to close down the water bodies. Now, the traditional methods usually identify or target either cyanotoxins like microcystins, or are focused on microscopy-based methods, which are cumbersome. They require extensive expertise, experts to look into the thousands of images, and from that, you can get a very one-dimensional information about a particular taxa that or some causing the harmful algal blooms, or a very specific type of cyanotoxin mixing all the other information which can be very diverse in nature. So we use the DNA sequencing to alleviate this issue and get a comprehensive profile of simultaneously identifying all the potential harmful algal bloom formers for a particular water ecosystem, and at the same time also identify their genetic potential to produce not just the cyanotoxins, but all the other compounds that can be related to the environment that they are where the blooms are forming. And this information can be integrated into the current strategies to control the cyanobacterial harmful algal blooms, and can basically alleviate the limited information associated issues with the traditional monitoring methods. And as Dr. Schellhorn has mentioned in his talk, our long-term goal is to create a portable, on-site, and low cost or cost-effective DNA capture devices, which are not specific to just one target or does not provide just a single information, but can be extended to hundreds of different bacterial pathogens or the DNA-based targets of interest, and can provide the information in real time without the use of any complicated equipments. Ideally, you can just use your phone to see whether you get any detect any signal in that water environment or not, right? And also, it would not require a full lab for you to perform the analysis on. So that makes it easier for the general population as well as the public health agencies to identify a lot of the bacteria or pathogens or the DNA-based targets at the same time on site without the use of complicated technologies. Thank you very much. That's the end of my talk, and I would welcome any questions that you would have at the end.
All right. So I'm the third speaker in this talk, and so my name is Ravi Selvaganapathy. I'm in engineering at McMaster and collaborate with Herb and others in terms of developing a comprehensive package for water quality monitoring. So Herb and Faizal talked about DNA-based analysis, and what I'm going to talk about is sensors that look at more elemental parameters, free chlorine, for example, in drinking water, pH of water, as well as phosphate, nitrates, and so on. So all of these require low-cost sensor fabrication methods. The current technologies that are out there. Let me see. Do you see the slides moving at all? No. Yes.
Yeah, that's working.
Yeah. Okay, good. All right. So the current microfabricated sensors that are available almost anywhere looks something like this, where you use conventional technology that is used in semiconductor manufacturing in order to manufacture sensors as well. And this is what I used to do like 20 years ago. I come from a semiconductor microfabrication background, and therefore it's easy to adapt those technologies here. However, sort of my central thesis and argument is that those technologies are an overkill, too expensive for making sensors where you don't need high density microscale components. You need sensors which are fairly large because they have to be deployed in the environment into a package kind of thing. So although these technologies provide high resolution, dense patterns, high throughput production, they still use expensive processes, materials, and this is one of the reasons why low-cost sensors have not been developed as much. So what we are interested in McMaster is to try to bring down the cost of fabrication of the sensors using scalable technologies that are out there. One of the technologies that we have used is basically a technique called xerography. It's a fancy name for using an automated cutter plotter, right? And so what you see in this picture is a cutter tool that is used for label cutting. So label cutting can be done in very high throughputs, in a scalable manner at a very low cost, and can give you precision these days of sub 100 micrometers, which is sufficient enough to form sensor elements. So we combine cutting with adhesive bonding. 3M, for example, makes thousands of adhesive films that can be used for this purpose, and then lamination in order to make our microfluidic sensors. Our approach is very simple. We use an automated tool that uses sort of 3D printing like technologies in order to engrave a film, a polymeric film on a substrate of any design that you can put into a computer. And our films are basically a sandwich of 3 layers. One is the polymer that we want to use, another is an adhesive, and then on the bottom is a carrier film. And then once we create one pattern, we create many patterns and then use alignment pins in order to align all of them in order to make microfluidic channels. Final step is lamination, which combines them using the adhesive in between to form very low cost, complex devices involving multiple materials. We can integrate filters, we can integrate electrodes and other purposes. And so using this, we've made a lot of microfluidic devices. One of the challenges in the past with these kinds of techniques have been integrating metal electrodes. So metal electrodes are difficult to integrate. Usually you use an expensive process like sputtering and so on in order to do this. We have gone to use metal foils and metal leaves for these purposes. Metal foils come in the range of 4 micrometers to 50 micrometers in thickness. Leaves come in the range of 100 to 500 nanometers in thickness. And these are very high quality metal films, but techniques to pattern them and integrate them don't exist at the moment. So we've developed these processes for doing that. So we use lamination to adhere our foil onto a substrate, cut defined patterns, and then basically using UV light, release these from the substrate, and then peel off whatever we don't want to create complex patterns. Using this, we can create heaters within, say, 5 minutes or so, electrodes for electrochemical and chemiresistive sensing very quickly, and without using any complex fabrication facility. So we can do it in our labs, in any facility that provides suitable equipment. What we've done is we've characterized this. We've shown that we can get sub-micrometer accuracy and precision in terms of manufacturing these devices. We made heaters and used this to integrate with microfluidic chips to heat certain elements of the chip versus the others. We've also done electroplating on these copper files in order to form gold electrodes, platinum electrodes, silver electrodes, and so on, and they show excellent electrochemical characteristics. Finally, we've also used metal leaves. So if you've eaten Indian sweets, for example, you would see a silver foil on top of some of these sweets. And these are made by age-old technologies. And the cost of the metal is essentially the spot price that you get in the market. So there's no extra processing cost for getting these large area, very precise, highly thin metal sheet-like structures. And they are very high quality. They are 99.99% gold or platinum or silver, and therefore they make excellent electrodes. What we have done is use the same techniques of xerography in order to make patterns like heaters and electrodes using these ultra-thin nanometer scale films over very large areas using the cutting technology that we have. And so these are some patterns that we've made. We've characterized what is the thinnest films that you can cut or electrodes that you can cut. And we've used silver, aluminum, copper, gold, palladium, and you can use a variety of materials that these leaves are available in. Using this, we have made flexible sensors. So this is a flexible electrochemical sensor that you can put onto wearable electronic type applications. And what we see is that despite bending and all of these things, even after 120 cycles of bending, the characteristics, electrochemical characteristics are exactly the same. We've also made chemi-resistive sensors. So this is where we use the same technology in order to make electrodes and then integrate carbon nanotubes or graphene using spray coating. And using this, we made free chlorine sensors that show good performance in these ranges. All of these technologies we are interested in transferring to applications. So we're working with a local company here in Canada, and they're partnering with many companies across the world in order to commercialize this technology for drinking water, swimming pool, and other applications. So with that, that's the end of my talk. Happy to answer any questions.
Thank you very much, Ravi, and thank you to all presenters. We're now moving into the Q&A portion of the webinar. So you're welcome. You're welcome to raise your hand and or put your questions in the Q&A section of the Teams webinar here.
Did I forget the form?
There's no M in between. It's Saleem.
So I'll just wait a few minutes here while people type in their questions and or raise their hand, and I can unmute you. Just going to check the chat as well. You could also place your questions there. This would be a first. No questions in a webinar. I think that's pretty amazing. Oh, there's a question that just came through in the chat. And the question is there a plan to combine DNA detection with microelectrode technology, and what approach in general would be used?
Maybe I can take that question. So definitely that was one of the projects that we had previously worked on, and the idea there was to use a combination of sample preparation electrically to lyse cells as well as extract the DNA into a chamber electrically, and then use some of the label-free techniques, either electrochemical or field effect transistor type technologies, in order to do the sensing. So that was one of the technologies that we'd worked in the past, but then after that, none of the companies were willing to go down that route, but that is one approach that we had pursued.
Thank you, Ravi. And there's a second question from Charles Delanoy. It says, for Ravi, that's for you directly. Can you talk more about how you're collaborating with Norgan Biotech?
That's a collaboration that Herb has pioneered with. Maybe Herb, you want to? You're muted though.
Oh, hi, Charles. We actually started a partnership with Norgan. The president is a McMaster grad, and he about 15 years ago, and we've done quite a bit of work with them, testing their kits and making sure that they are robust and can protect the communities that we're interested in relative to those made by other manufacturers. And we've worked on a couple of projects that are kind of proprietary, but they've been a long-time supporter of our work, and they were heavily involved in wastewater analysis for COVID for many American universities because their proprietary kits detect pieces of DNA that others cannot. I'd like to come back just to the previous question briefly. We are, of course, working with Ravi on DNA detection, and Faizan mentioned that we have these unique conserved signature protein genes which have been validated through about 150 publications from the Gupta lab, who is a founding member of the MacWater group and a close collaborator. And this is something that has revolutionized bacterial taxonomy to help us understand the relationship between evolutionary relationship between groups of bacteria, including pathogens. And as you can imagine, if it's good enough for doing that, then it's great potential, has great potential for diagnostic technology. And that's the case. That's what we've just published several papers on that, specifically for different water-related microorganisms. And the idea would be to combine that information into a DNA chip in conjunction with Ravi that would serve several purposes. So I think there are aspects that are unique to McMaster, the interdisciplinary nature of our research, but also this made in McMaster technology, as Faizan described it, is really unique to McMaster. There are Rad Gupta has dominated this field like nobody else, and now he's after 25 years, he's become one of the world's foremost authorities on this.
Thank you for that, Herb. Again, if you have questions, please either post them in the chat, use the Q&A function, or raise your hand and I can unmute you. Give everybody a minute.
I should mention, if there's time, just that people can contact myself or Ravi or Faizan. I understand I have to correct his email a little bit, but we are interested in identifying other problems, and we have one of our partners is Waterloo Biofilters that we've worked on for many years. A great state-of-the-art septic treatment company that has innovative technology and is now expanding into the U.S. and we like to see that. It's headed by a former Cisco executive now, and so that is really been an interesting development because now it came down to McMaster to expand the research partnership, and that's been very helpful. We've obtained several partnership grants with their support that didn't cost the company any money at all, but it helped leverage federal funding for translational research that they're interested in.
I can see a question in the Q&A section from Vince Pelleggi of MECP. Have any applications for compliance purposes been done? For example, WWT wastewater treatment final effluents need to comply with low E. coli, low TP, low TAN, low BOD, low TSS on a monthly average basis to meet low milligrams per liter levels. Often we find municipal wastewater treatment operators, plant owners challenged with daily or more frequent sampling, analysis, and reporting to the MECP.
Well, yeah, and that's a really interesting question. We just received an email for collaboration from a waste treatment operator in Australia who has been using DNA technology, and they want help with the analysis of data. Now, coming back to Ontario, septic manufacturers have the same issues. They have to report BOD, total solids reduction, total E. coli, and measure parameters. So we were engaged in a partnership grant to do a test deployment of technology at the Minden municipal waste treatment plant over the last 3 years, and we've combined DNA data with their standard traditional effluent monitoring. This can be applied to large wastewater operations, which we haven't worked with large wastewater groups very much, and that's we have done a little bit, but I think that's something that we have to do as a sort of formal interaction. And the MECP hasn't made that a priority for us. We've worked under contract to the MECP on two projects. The Enterococcus was funded, and the algal bloom project was funded by the MECP. Both were matched by NSERC Alliance, so that was pretty good funding. And we published several papers in each case. So we would be interested in working with large wastewater operations, but many of those have their own analysis labs too. Hamilton and Niagara, for instance, have their own environmental services lab that conduct analyses. And we have had contact with Niagara in the past through the Water SMITE program. We've dealt with their we've collaborated with their environmental services division, which is headed by a McMaster grad, actually. So these things help.
If I may add, I think one of the things that we are working, maybe not in the wastewater area, but in terms of food quality analysis is, so I'm part of a startup company that does automated detection of food pathogens in meat. So problems are similar. You need some incubation of this to detect viable species that are present, and then basically do PCR type analysis. And there, what we are doing is taking the skilled work out of the point of use and automating the entire operation, and also using AI technology to analyze this data and interpret it based on all the other parameters. So all of these parameters and tests could potentially be automated because the technology is getting cheaper and cheaper over the past 2 years or so. And that is being sort of sought after in the food industry, primarily because they have to meet regulations and so on. And it is possible that similar kind of things can be applied in the wastewater area as well.
Thank you very much, Ravi. There's a question in the chat that is for the OWC, actually. Is there funding through OWC and or the ability to help with matchmaking between labs' capabilities and local industry needs and interests? At the moment, we don't have funding we can make available, although we're in hot pursuit of funding at all times and are trying to submit a proposal for new sector-wide funding that can help advance innovations in the water space. So stay tuned for that. But in terms of the ability to help with matchmaking, there may be a possibility for us to help with that. Charles, I will reach out to you after the event to connect with you and learn a little bit more about what your needs are, and then we can discuss further about how we might be able to help. Then we have another comment, just a comment by Vince in the chat that says, automation would be very useful. Thank you. Thanks for your questions and comments, Vince. Are there any further questions or comments that anybody would like to make to the group here today? Give everybody another minute. There's a question that just came through by Sonia Hassini. Is there any work done or interest for OWC in monitoring stormwater management control in ponds or low impact development? Is there any work? And the question is, the OWC is the sort of administrative head of all our post-secondary institutions and the research capacity that exists within those institutions. So we don't engage directly in any of the research. Of course, our partners do. I'm not sure, Sonia. We can follow up after to be. Perhaps you can clarify a little bit, and you can. I can shoot you an email after to see what it is you're looking to find out about the OWC. Unless this question was actually for Herb, Ravi, or Faizan.
Yeah, we're just waiting for money from OWC.
I know. Us too, Herb.
Or anybody else that has funding. I think it is important.
Yes.
And
we are always in pursuit of new funding and convincing the government how important it is to continue funding the water sector and the innovation that it's definitely needed going forward, and now needed today and for the future. Yes, and we need many advocates in our corner to continue doing that, to bring water as a priority, as a cross-sector priority for all of the national and provincial interests that exist in our government, as it really crosses all of the priorities and is needed for all the priorities and to achieve all of the goals of our current federal government, our current provincial government. Okay, I'm just going to check one more time here. There's no more questions in the Q&A, and there are no more further questions in the chat. I think we'll end it here. Thank you very much to all of our presenters here today, Ravi, Faizan, and Herb. Thank you very much for being here and your insightful presentations. I hope everybody enjoyed this webinar. You're welcome to get in touch with the Ontario Water Consortium or with the MacWater Diagnostic Initiative directly through our website, ontariowater.ca. We can always connect you directly to Herb as well. Have a great rest of your afternoon, everybody.
Thank you, everyone, for attending.
Yeah, thank you very much for the opportunity to talk about McWater and partnerships. And you're all invited to our meeting. Just send me a note next year, 2026.
Sounds good, Herb. Thank you very much.
All right. See you, everyone. Bye.
Bye now.